In the last class, in one of the last classes before I went on leave, I was discussing about breast cancer, right? So we discussed about the cycle, the we discussed about the cycle through which the breast goes through during the different developmental stages. One second, yes. So we also discussed about how it is natural, what should have been the natural course, what is the unnatural course as humans because of the modern day living, the changes that we have. And I remember I did discuss something in this way about what humans select for um, bigger, larger breasts or not. So the discussion there for me was about evolution. In that sense, the point that I was trying to make was, or trying to discuss with you about, is it natural selection or sexual selection? I hope you know what is the uh, difference within those. Natural selection, at least, is the nature imposing its restrictions on whether something will survive or not, and that is dependent only. That is basically if an individual is able to survive the harshness of the nature, procure food and survive or not. If when there is nature, and those that are surviving larger for longer duration are likely to, or it is simply the survival of the fittest, right? In sexual selection, it is the individual, a male selecting a female or female selecting a male. Usually the males are the ones that are selected by the females. Okay, so in the why I'm bringing this up again is I have been thinking about it, and I wanted to try to address this as well. Is there selection acting upon females? I mean, if it is sexual selection, is it acting? Natural selection acts on all individuals. There is no question about it. The question that we have is whether are the males selected or are the females selected? Say if I will try to compare what we are closest to, say the primates or apes, for example. The alpha, the alpha male gets all the mating rights, and all the females will be impregnated. So the selection there is happening on the males, right? Females do not have a choice in this case, because whoever is the alpha gets all the writing mates. I mean, there is stiff competition from other males, but it is about the male. The reproductive rights are with the male that is the dominant one. Okay, so whether the larger breast was selected or not, then is likely to be the natural selection might be the reason for it. Say, for example, those with larger breast may be able to give milk for longer time, longer duration, and more milk, which means the child is fed for a longer duration, which means that child will grow to adulthood and then reproduce. So that could be the reason for it. Okay. Every biological thing we have to discuss in the light of evolution. That's always, there is a nice quote also by someone, I forgot the name right now, but all of biology makes sense only in the light of evolution. That's what we should say. Okay, so we have discussed about milk production, and we were also discussing about the stem cells, I think. The may eventually, or sometimes they may form cysts. It is a little bit common that many women do have lumps in their breast based on, it depends on infections and many things, but sometimes they might be lumps which might keep on growing, and then they would grow the that is the tumor. Okay, and it can grow in different grades like 1, 2, 3, and eventually it will undergo metastasis, and then it becomes fatal to the individual because tumors will grow in different other places. Breast as such is not an essential organ for that individual, isn't it? So when such things occur, these are I was also discussing about the metastasis and the secondary sites of metastasis. That means where else, say for example, a tumor that has formed in the breast may relocate, of the cells will go and form different tumors in different other parts of the body. Here is a table. It's not complete. It is incomplete. So let's see if a breast cancer types, these cells, if they have moved out into the bloodstream, they are actually theoretically they're accessible to every part of the body, but they somehow cannot probably make the tumors in other tissues. And then some of the sites are where they do survive and make tumors are lungs, liver, and bones. Similarly, if it is a colon cancer, they might go into lungs, liver, peritoneum, and so on. And I hope you can see those things. What I want you to focus on is you will find that lungs is kind of everywhere. Majority of the tumors can make tumors in the lungs, right? Almost all of them, which is, I think, in at one point I was discussing why lungs are a good site for secondary tumors, because they have space to start with, and then they have nutrition, or there is complete blood supply. So space is available through blood, you will get the nutrition, and also it is anyway in the O2 in the lungs, so it is well aerated. So I think that's why lungs are a most likely secondary sites for tumorigenesis. Okay, there are multiple different types of breast cancers. We don't have to really mug these up. It depends on which tissue, whether it is happening in the ducts or whether it is happening in the lobules. These are the different types of breast cancers. I was also, in some years ago, I learned that even men can also get breast cancer. It's a very, very rare situation, whereas as opposed to what it was for women, breast cancer is one of the um, high incidence in women. Like prostate cancer is in with men, and specifically, right? So how it usually manifests is as a tumor grows. There could be some misformations within the breast, where they would call it some dimples or some changes. One should not be alarmed just because there is a dimple. Not always it means that, but if it is persistent and there are many other symptoms, there are several tutorials on YouTube also, where you can try to see how to do self-examination and then decide whether you can meet doctor or not. Medical advice is always the best in any case. So if the doctor suspects, then they may ask for a radiogram, where you can see that the tumors they can be found out. And the solution for it is to remove these majority of the, or mastectomy. I think that's what is called as, where they remove the breast. And for a woman, breast is kind of um, an organ that is looked for at a point of attraction. So removing those is actually usually a psychological trauma to the individual. So some of them, this is a double mastectomy, where both the breasts are removed. And say if somebody wants to go to the beach and it'll be a cause of concern for that individual, because it's an abnormal not to have, if for a female not to have breasts. So they do something like this, cover it up with tattoos. That's just I'm trying to show it is in a good spirit, that you can, when survival is more important than other things can be managed, right? Existence first. Existence is the essence. Okay. So the basal the what we are trying to talk about right now is cancers, and the breast cancer, and we will eventually get into the cancer stem cells, and that is what we are trying to see. So there are several discussions or debates whether the breast cancer stem cell, what are its properties and other things like that. Some people believe that it is like basal like phenotype, whereas others will say no, it's not. Maybe it's lobular. I mean, the basal like phenotype, I hope you remember the basal cell. And then we have these cells that actually produce the thing, like in the prostate gland, for example. These are the basal line, the outside one, right? This is basal. So where do these cancers arise is an important question. Whether do they have a phenotype like the luminar one, or do they have something like the basal cell like? So at least um, we will discuss about BRCA in a short while, but this is what people believe it, or what people have found is that majority of the phenotypes, at least of those that are BRCA1 mutants, they are something they show like basal like phenotype. So there are other cell surface markers. We don't have to really bother about them at this point, because we are concerned, we are reading these parts as a part of cancer stem cells. Okay, this is stem cell biology course. So how do we identify? And this is I gave you a question on CAA too, so I hope you will be able to relate it. It's also a kind of revision in that sense. How do we identify breast cancer stem cells? And if we are going to use cell surface markers, and for that we make a single cell suspension, and then we can do cell sorting based on lineage minus, whether they are present or not. Once we have made lin plus or lin minus, and then we can test for all those. They would have tested for whether lin plus cells, are they causing tumors or not. What they found is they do not cause tumors. They checked whether lin minus, can it cause tumors or not, and tumorigenicity is observed. And then they went to further make based on CD44, for example. And then you could also, after CD44, they have also tried for CD24. Okay, so if I want to represent this one, it would be lin minus CD44 plus. Okay, assume this is plus and here is minus. CD, this is plus and this is minus, just theoretically. Okay, CD24 minus, that would have been its cell surface markers phenotype within the range of what the experiments have been conducted. Okay, let's see what they have done. So they first did with lin, and then they have done sorting based on CD44, 24, and ESA. So there are three cell lines, three cell types. They have 4 cell types they have got: 44 plus, 24 minus, lin minus. All of them are lin minus, so I don't want to mention that. And then they have CD44 plus, 24 plus, right? And then 44 plus, 24 minus, and ESA plus. And here it is ESA minus. So each of them they have taken and transplanted into nod or skid mice near the mammary pads and see if they are giving any tumors or not. So based on the results, this is not giving tumors. This is are giving tumors, and these are not giving tumors. The unsorted cells, the single cell suspension, they are also giving tumors. What do you think is the cancer stem cell phenotype here? Anybody?
Sir, can you repeat, sir? I didn't listen.
Okay, I will repeat. I hope you. These are the cells taken and given into the tumors like this. Okay, and these are the results that are found. That means in there are some cases where the tumors have not found. There are some cases where the tumors have found. This is not no tumor here, and here also no tumor. We don't have to bother about it. There are two phenotypes which have shown tumorigenicity. That is this one, CD44 plus 24 minus lin minus, and this one, CD44 plus 24 minus ESA plus. Right? How many cells is this? This is 1000 cells. Here it is about 200 cells. So the cancer stem cell phenotype, what do you think it is from the existing data?
CD44 plus and CD44 plus CD24 minus.
And
Sir, with or
ESA,
without ESA also?
ESA plus. This requires only 200 cells to give rise to a tumor. So basically, CD44 plus 24 minus, these two are almost same, right? Except this has ESA plus and ESA minus cells also, both together, right? In this part, they have segregated the same phenotype as this in based on ESA plus and ESA minus. And what did they find? They found that ESA plus cells actually have the tumorigenicity capacity, whereas ESA minus cells are not giving any tumors. Okay, I hope you understood this part. So this is the phenotype that is the um, at least by the time of publication, breast cancer stem cell cell surface markers are CD44 plus, 24 minus, ESA plus, and of course lin minus. Okay. Yeah, that was the question. So we learned about embroid bodies and tumor spheres. Very similarly, we can also get mammospheres. These are, if we take a normal breast tissue and then we make into single cell suspension, and then we have removed the extracellular matrix, and if we place them into a culture dish and into non-adherent suspension, something similar to that of embroid bodies, like hanging rope method, they grow into something like a mammary gland itself, a small gland lobule, and those are referred to as mammospheres. The same thing if we have taken from a breast cancer tissue, they also form similarly, similar to that of what is mammosphere, similar to that of mammosphere, but this one would have, obviously, will have cancerous cells. Okay, you can do serial passaging and other stuff to determine the other characteristics of self renewal and tumorigenicity and differentiation. This is basically this saying this the same thing. Okay, with pictures. So when I was discussing about the what are the breast cancer stem cells phenotypes, it does seem to be having several features that are bordering that of mesenchymal stem cell like, or other adult stem cell like, and also basal like features. based on the type of signaling pathways that are present in that. Okay, we don't have to worry about it. So long time ago, when I was started to teach this course, then at that time, because the authors of the paper say it's basal-like stem cells, and then there was a publication on it, I didn't follow it up. Like BRCA-like basal-like breast BRCA1 basal-like breast cancers originate from luminal cells. Luminal epithelial progenitors are not from basal stem cells. Okay, so I hope you remember that we have been always having this discussion about these are the stem cells. They would give rise to transit amplifying cells, and then they would eventually give rise to the functional cells, right? We were always discussing about what is the origin of the cancer stem cell. Is it from the stem cell, adult stem cell that is present here, or is it from the transit amplifying cells, or is it a mutation in the completely differentiated cells that give rise to the cancer stem cell? This paper, at least based on the findings at that time, what they found is that they usually arise from the luminal epithelial progenitor cells. Okay, just to put into perspective, we don't have to take that this is for sure the only way. Maybe in the future, with better techniques and understandings, we will be able to find out more or better answers. Right now, I want you to have, when it is your turn to look at some data, you should be able to see it in proper ways and put it into perspective. Okay, we have been discussing about BRCA, and what is BRCA is something we need to discuss. It is an important something like a tumor suppressor gene. It has important functions in chromatin remodeling, in DNA surveillance, ATR, ATM. If there are mutations in the DNA, it gets activated or it is targeted, and then it allows the repair of the DNA, gene expression and signaling, and some protein degradation as well. So it is kind of central one in this one. I hope you know who she is, right? Angelina Jolie. So she and her mother and her grandmother, all of them have this BRCA mutation. And because of some things, probably she had suspicion, and then she went for testing whether she has the mutation, she has inherited the mutation from her mother. And it was found out that she has this BRCA mutation, and she underwent mastectomy, and of course probably there will be implants and other stuff, but she has got there are two possibilities. I think it is uterine cancer or ovarian cancer is one possibility, where they get rid of the ovaries or the uterus. I can't remember what is exactly in her case, but also the breast. So she got these removed to prevent future attacks of cancer. The same thing, the many people are also trying to do the similar things by getting genetic tests so that they can live longer and better. So as I was discussing, the BRCA mutation BRCA protein is important regulator in several of these cases. Check 1, check 2, and these are the checkpoint regulators. If there is any mutation or so, then the BRCA1 is activated, and then it regulates several of the things. Cell cycles stoppage, and then it will initiate several other things so that the repair mechanisms or repair of these genes through ATM and other things. So I hope you understand that the BRCA1 is in the center of many events It prevents cell cycle progression. It will allow the transcriptional responses to, that is through for repairing the DNA damage and things like that. And also chromatin remodeling and DNA repair, many other things like that. What would happen if BRCA is mutated? Then certainly the functions, the downstream functions are going to be affected, and that is exactly what happens. So there is one more thing that we also want to discuss is autoactivation. Interleukin 6, it activates or notch 3, the signaling pathway, which also crosstalks with RAS and EGFR, and then they are activating the ERK, that is, it comes in the RAS pathway, and ERK activates JAG1 and carbonic anhydrase 9. We will see some of them, if not all of it. And all these things together will allow the production of more IL-6. It is kind of positive feedback, right? So IL-6 is activating um, notch signaling, which will allow the survival, proliferation, and differentiation properties. Then we have erythrocyte-like growth factor receptor that is activated, which is something important for mammoth spear formation and survival. And then we have the carbonic anhydrase, which is important for hypoxia survival. We will have a brief look at them. If we take a cross section of a tumor, okay, this is where the blood supply, blood vessel is. In the area where the blood is present, we will have high concentration of O2 and also nutrition. Here we are more concerned about the oxygen. As the distance progresses from that of the blood vessel to the periphery, we will have hypoxic conditions. There will be less and less of oxygen, and because of which we will have increased H plus ions. That is, we will have low pH here, acidic conditions in the periphery compared to that of the what is happening near the blood vessel. Okay, that is because of two factors that will. First thing is, without oxygen, survival is difficult, and that is the Warburg effect that you might see. You might The cells will rely more on the glycolysis than on Krebs cycle. Glycolysis is an anaerobic activity, which means that it doesn't require oxygen to happen. But the problem is, one glucose molecule will give about 2 ATP molecules only that are directly usable. And we also will have the production of lactic acid, which is going to increase the acidity in these locations, right? So when beyond that, in this region, the tissue is dying because there is absolutely no supply of oxygen as well as probably nutrition also. That tissue is undergoing necrosis. So here is, I like to present this slide and how a tumor, as if it has self-regulatory activities and survival strategies. It has some of the tumor cells make a kind of symbiosis. It's referred to as the metabolic symbiosis here. So there are cells. This cell here is not receiving oxygen, no oxygen or hypoxic condition. And this cell, the bottom one, is having aerobic conditions. So the aerobic cell can undergo or carry out electron transport chain, whereas the top one, the hypoxic cancer cell, cannot. So what it does, what should happen is, in this case, it should increase the glycolysis, reduce the TCA cycle. But the problem is, it will give out lactic acid, and only 2 ATP are produced. That means it has to take more glucose into it, degrade all of it into lactate, and derive whatever ATP comes up, and then pump out the lactic acid. So, what is the metabolism? What is um, the tool here? Sorry, I will just want to say. Oops, sorry. Okay. Mm, where is it? Sorry. Anyway, I hope you will be able to see. I did something. Just give me a minute. Hmm. I'll just take that and I will stop. Okay. Yes. So the metabolic symbiosis that I was talking about here is that the the top cell, the hypoxic cancer cell, would want to take as much as glucose possible inside. So it has to have more transporters, GLUT1 transporters, to take up the glucose, perform or carry out glycolysis, produce the ATP, and the pyruvate that is produced will convert it into lactate, and it should also have these exporters or efflux pumps to send out lactate. The aerobic cell cannot doesn't need glucose. It can instead it should be able to take up this lactate, the MCT, other exporters or pumps, and then take the lactate, convert it into pyruvate, and then send it for the electron transport chain and produce large quantities of ATP. So normally we would have seen that in the cytoplasm, glycolysis would have occurred, and then the pyruvate would have been sent into the mitochondria, where ETC would have, electron transport chain would happen. ATP is used up by the cell. Everything would have happened in one cell. But now, because of the way the cells are within the tumor, some cells are receiving oxygen and some cells are not, and then we can still see that it's a beautiful arrangement that they have made to um, make the best out of the available glucose, make the best of the available glucose with the limitation of oxygen. Right, there is oxygen limitation in the tumor. There is assumed supply of glucose is fine, and in that case, how does this metabolic symbiosis happen? And the last is there is one more that is called the carbonic anhydrase. The it is the as the as it is carrying out metabolism, we would have the pH. We would have had lactate produced, and we need to somehow, this cell should be able to survive in these acidic conditions. First thing is that how can a cell survive within the acidic conditions? That means the cell has to, inside the cell there should be homeostasis, right? If there are no there is homeostasis is important, because all the enzymes that they work are evolved or refined or optimized for a particular pH and other physiological conditions. Unless the cell is able to maintain those conditions, the cell cannot perform the metabolic activities. So how does that um, homeostasis, how can homeostasis be maintained is what we are discussing about here. So there are two things: anion exchanger and carbonic anhydrase. Anion exchanger and Ca9, carbonic anhydrase 9. So what we will do, we'll think is, so assume that bicarbonate CO2 is here. It will go inside. Okay. The point is we should be able to get this proton out of the cell, right? That is where we are able to this is against the gradient in a way, because you already have high H plus ion concentration in the extracellular side, and we there is a possibility we should be able to send these H plus additional ones out as well, right, to keep the cytoplasmic pH properly. So the H plus will combine with HCO3 and will give rise to CO2 plus H2O, carbon dioxide and water. This carbon dioxide can diffuse out of the cell membrane, and water will also. And there it might say the carbon dioxide will can react with water and form the carbonate ion here and release the H plus. So I just want you to focus on what is happening to this H plus. H plus combined here, and it is um, it has formed water which can move out, and there it is dissociating, and that activity is performed by carbonic anhydrase. Okay, and yes, the anion exchanger that we have here will take this ion here back in exchange for chloride ions. Okay, and this cycle can keep on going as it is doing. More and more hydrogen ions are removed from the cytoplasm and they are released into the extracellular site. That is how the cell, although it is in the acidic conditions, can maintain its pH so that it's a physiological pH so that it can function and survive in those conditions. Okay, I will stop here. I do have a few things to discuss, and we also have to once review how many more chapters are there to finish, because I think in the next class I will take up a few parts of this and then I will stop. This is also a nice thing. Why I did have these in the PPT is for us to understand the behavior of cancer. It is one cell has got mutations, and then it is almost organizing itself like a parasite in the body and making adjustments with the based on the conditions it is receiving, like hypoxic conditions, and if there is no blood supply. Okay, if there is hypoxic condition, the other one is to increase the blood supply. So there is, if we increase the blood supply, then there is more amount of oxygen available. So for that, it will allow it will try to trigger the body to make more blood vessels, that is the angiogenesis. Maybe in the next class I will discuss this one slide, and then we will move on with the rest of the things. Okay. Any questions? Do you have any questions from yesterday's class, yesterday's exam? Okay, then we'll see you in the next class. That's 2 days holidays. Wish you a very happy Diwali. Have fun, make good use of it, but enjoy equally.
Happy Diwali and thank you, sir.
Thank you,
Yes,
sir.
thank you. Thank you. Have fun.
Sir?
Yes.
Sir, regarding IAS fellowship, sir, if
Uh-huh.
I can give you details, sir. I'm applying for health science, sir.
Okay. Yeah, you can give. Why
Okay,
health
sir.
science?
So I'm actually interested in it. It's like health science and medicine they are given
No,
medicine.
why I'm asking is think about whether the number of openings are more or less.
Okay, sir.
Okay. And think about the probability, but you can certainly apply anything. Yeah, that's true.
Okay, sir. Okay,
Yeah,
sir.
yeah.
So it's actually I like to work in lab based on medicine and health, sir. So I just thought of applying it.
Yeah, sure. Okay.
Yes, sir.
Yes,
Thank you,
fine.
sir. Happy Diwali, sir.
Yeah, happy Diwali. Have fun.
Yes, sir. Thank you.